Hello, today in Nagarn. Today we're going to be solving Feb-March 2023, Paper 1, Variant 2. We are currently offering free Topic 1 notes. All you have to do is fill in the form in the description box down below. Question number 1. The photomicrograph shows cells from a human blood smear. Which calculations shows correct method to calculate the actual diameter of the neutrophil shown in the photomicrograph in micrometers? So as we all know, the equation for magnification is IM. And we are trying to calculate the actual diameter. Therefore, it's the image size divided by the magnification. So the image size is going to be the distance from P to Q. And in order to convert millimeters to micrometers, we must multiply by 1000. Therefore, this is correct. And divide that answer by the magnification to obtain the actual diameter. Therefore, the answer is going to be A. Question number 2. A student calibrated an eyepiece graticule using a stage micrometer. What is the best estimate for the diameter of the alveolus? First of all, we must define a few terms. What is an eyepiece graticule and what is a stage micrometer? So, an eyepiece graticule is a small scale that is placed in a microscope eyepiece. And it is a scale with specific divisions and without known dimensions. So it's only a ruler divided into equal lengths. Contrasting a stage micrometer, which is a scale with known dimensions and is placed on the microscope slide. And it is with known dimensions. So first of all, we must start by calibrating that eyepiece graticule at that same magnification. So each division of the stage micrometer was 0.01. With a 10 magnification objective lens, 10 eyepiece graticule units match 10 divisions on the stage micrometer. Therefore, it's 10 and 10. So what we must do is we multiply 0.01 by 10 to obtain the length of 10 eyepiece graticule units at a magnification of times 10. So let's just assume that these are 10 eyepiece graticule units. Therefore, we get 0.1 millimeters. So 10 eyepiece graticules equals 0.1 millimeters at a magnification of times 10. Now, we want to know that same calibration at times 40 magnification. We know at times 40 magnification that we are zooming in deeper. So what we must do right now is that we divide 0.1 by 4, because we just said that 10 eyepiece graticules at that magnification is going to be 0.1 millimeters. Therefore, what we are doing is we're dividing 0.1 by 4, which would give us 0.025. So now, 10 eyepiece graticule units at a magnification of times 40 is going to be 0.025 millimeters. Now, we want to know what 96 eyepiece graticule units would be. So what do? Is we divide 0.025 by 10 first, and then multiply that answer by 96. So we get 0.24. So first of all, we're dividing by 10 and then we're multiplying by 96 in order to find 96 eyepiece graticule units, which will give us 0.24 millimeters. And now we need to convert 0.24 millimeters to micrometers, and we have to do that by multiplying by 1000. So if we multiply by 1000, we get 240 micrometers, and the answer is going to be C. Question number 3. Which statements are correct for chloroplasts and also mitochondria? So chloroplasts and mitochondria have prokaryotic origins. So this is our assumption for this question. 1. They contain 80 S ribosomes. This is incorrect because they have prokaryotic origins. They have 70 S ribosomes. 2. They can transcribe their circular DNA. This is correct. They do have circular DNA, which they are able to transcribe since they have prokaryotic origins. And that makes them able to translate the mRNA. So this is also correct. Now 4. They're enclosed by double membranes. Yes, both the chloroplasts and mitochondria have double membranes. If we would draw the mitochondria, for example, it does need a double membrane. Therefore, the answer is going to be C. Question number 4. The electron micrograph shows some cells from a root. Which cell structure is not usually found in cells from a root? So, the function of the root is the active uptake of minerals and the passive uptake of water. Therefore, it is not common for root cells to have chloroplasts, since chloroplasts are found in the leaf where photosynthesis takes place. Therefore, the answer is going to be B. Question number 5. Dialysis tubing is an artificial partially permeable membrane with pore sizes of approximately 2.5 nanometers. Glucose molecules have a diameter of 1.5 nanometers and can pass through the pores in the membrane. What else can pass through the pores? First of all, you must know what the average size of prokaryotic cells are and the average size of eukaryotic cells. And prokaryotic cells range from 0.1 to 5 micrometers, and eukaryotic cells range from 10 to 100 micrometers. Ribosomes range from 20 to 25 nanometers, so 20 if it's a prokaryotic cell and 25 nanometers if it's a eukaryotic cell. And here it's telling us that the diameter of glucose is to 1.5 nanometers. And we know that fructose is a monosaccharide, same as glucose. So we are assuming that they approximately have the same length, which is going to be 1.5 nanometers. Now, the size of the pores are 2.5 nanometers. Here we're looking for structures that are less than that size. Therefore, what we only see here is 1.5 nanometers, which is fructose, and the answer is going to be question number 6. What is present in all viruses? A. Ribose. So ribose are present in RNA viruses, also known as retroviruses. However, viruses could be DNA or RNA. Therefore, this is not always the case. B. Deoxyribose. So deoxyribose are found in DNA viruses, and we just said that there are DNA and RNA viruses. Therefore, this is not always correct. Adenine. Adenine is found in the base sequence of both DNA and RNA. Therefore, this is correct. Thymine. So, thymine is only found in DNA viruses. However, in RNA viruses, this is substituted with uracil. Therefore, the only correct answer is going to be question number 7. To estimate the concentration of glucose in an unknown solution, equal volumes of a range of known concentrations of glucose were each mixed with the same excess volume of Benedict's solution. After mixing, the solutions were placed in a thermostatically controlled water bath at 90 degrees for 3 minutes. So this is a control variable. The unknown solution was then treated in the same way and the colors of the known and unknown solutions compared. This is the dependent variable or the measure variable. What is the independent variable in this procedure? So the independent variable is the change variable. So let's see the suggestions that we have here. A. Concentration of glucose. Here, the question states unknown solution, so glucose concentration in an unknown solution. And we are measuring the glucose concentrations by the color of the Benedict solution. Therefore, the change variable, or the independent variable in this case, is going to be the glucose. Now let's go to B. Final color of these solutions. So the color of these solutions is what we use to measure the change. Therefore, this is the dependent or the measure variable, so this is incorrect. C. Temperature of the water bath. As stated here, controlled water bath at 90 degrees Celsius. This is a control variable. Therefore, this is incorrect. D. Volume of glucose solutions. So the volume of glucose solutions are constant. Therefore, this is a control variable, and the answer is going to be A. Question number 8. Which statements are correct for amylose and also amylopectin? So let's start by defining our terms. Amylose is a polysaccharide formed by the condensation of alpha-glucose monomers by alpha-1 to 4 glycosidic bonds, contrasting amylopectin. It's also a polysaccharide joined by the condensation of alpha-glucose monomers by both alpha-1 to 4 glycosidic bonds and alpha-1 to 6 glycosidic bonds. Therefore, amylopectin is branched and it's shorter. So let's see the suggestions that we have here. 1. They are carbohydrate molecules. This is correct because they are formed by the condensation of alpha glucose monomers. Therefore, this is correct. 2. They are formed by the condensation reactions. This is also correct. 3. They are linear molecules. Only amylose is a linear molecule because it's only formed by alpha 1 to 4 glycosidic bonds. However, this is incorrect because amylopectin is a branched molecule formed by alpha 1 to 6 glycosidic bonds. So these form the branches. Therefore, 3 is incorrect. 4. They contain alpha 1 to 4 glycosidic bonds. Yes, this is correct. Therefore, the answer is going to be B. Question number 9. Which statement is a correct comparison between saturated triglyceride molecules and unsaturated triglyceride molecules of approximately the same molecular masses? So, first of all, a triglyceride molecule is a molecule formed by glycerol, 1 glycerol, and 3 fatty acids. And saturated triglyceride does not contain carbon double bonds, and unsaturated contains carbon double bonds. So let's see the suggestions that we have here. A. Unsaturated triglycerides have more double bonds and fewer hydrogen atoms than saturated triglycerides. These 2 points are correct. The reason for that is because we said unsaturated triglycerides have double bonds and they do have fewer hydrogen atoms. Why is that? It's because when we remove a carbon single bond and form a double bond, what we are doing is that we must remove the hydrogen molecules because carbon can only form 4 covalent bonds. Therefore, there are going to be fewer hydrogen atoms by introducing carbon double bonds. Therefore, the answer is going to be A. Number 10, the diagram represents a molecule from a cell surface membrane. Which description of the labels is correct? First of all, you must figure out that this is a phospholipid molecule and it is amphipathic. Therefore, it means it has both a hydrophilic head and hydrophobic tail. And it consists of a molecule of glycerol, true fatty acids, and 1 phosphate, inorganic phosphate. So, due to the inorganic phosphate, there is a hydrophilic head and hydrophobic tail. Now, hydrophilic means it is water soluble and can form bonds with water molecules. Hydrophobic means it is not water soluble. Now, let's see the suggestions that we have here. A. Fatty acid at the hydrophilic end of the molecule. So we just said that it is hydrophobic, so this is incorrect. B. Hydrophobic end of the triglyceride molecule. This is a phospholipid molecule, therefore this is incorrect. C. Hydrophobic end of the glycerol molecule. So glycerol molecule has a phosphate group bound to it, so the hydrophilic head, therefore this is incorrect. It is hydrophilic. D. Phosphate group at the hydrophilic end of the molecule. This is correct. The phosphate can form bonds with water molecules, which makes it hydrophilic. Therefore, the answer is going to be D. Question number 11. Which row correctly shows levels of protein structure that can be held together by each type of interaction? Hydrogen bonds. So, there are 2 key rules. Hydrogen bonds form between the peptide bonds of the secondary structure and the R groups of proteins in the tertiary structure. So let's explain further. The peptide bonds are able to form hydrogen bonds with each other at the secondary structure. And in the tertiary structure, there are the R groups of the amino acid, which can therefore form hydrogen bonds with other R groups. So that's the key difference. Therefore, C and D are correct so far. Now, hydrophobic interactions. Hydrophobic interactions only form between the R groups in the tertiary structure. Therefore, it's tertiary structure. Now, covalent bonds. So, covalent bonds, an example for it would be peptide bonds in the primary structure, and it could also be disulfide bonds in the tertiary structure. These bonds are incredibly strong and they are hard to break. Therefore, primary and tertiary structure are correct, and the answer is going to be C. Question number 12. Which molecules contain at least 3 double bonds? So, in addressing this type of question, you have to know some key points. So, in a triglyceride molecule, we know it's formed of glycerol and 3 fatty acids. We know that fatty acids contain a carboxylic group consisting of a double bond between the carbon and oxygen. Therefore, it must contain at least 3 double bonds in each fatty acid. Therefore, triglyceride is correct. Now for collagen. We know that collagen is a protein. So let's see the composition of a peptide bond. So this is a peptide bond. We could clearly see here a double bond between the carbon and an oxygen atom. Therefore, collagen also contains at least 3 double bonds. Same with hemoglobin. Hemoglobin contains globin, which is a polypeptide, which is also a protein, so must contain also at least 3 double bonds. Therefore, the answer is going to be D. 13. Which diagram correctly shows hydrogen bonding between 2 water molecules? First of all, in describing hydrogen bonding, we must always use this sign to describe the charges. Therefore, A and D are incorrect. Now, we must know that the charge on this oxygen ion is going to be a negative charge, and the charge on hydrogen is always positive. Therefore, the only correct answer must be B. Question number 14. Which statement describes an example of an extracellular enzyme? An extracellular enzyme is an enzyme that operates outside the cell. So let's see the suggestions that we have here. A. Amylase in saliva is an enzyme that catalyzes the breakdown of starch in the mouth. Yes, amylase is in saliva, therefore makes it an extracellular, therefore this is correct. B. Carbonic anhydrase is an enzyme that helps in the transport of carbon dioxide in blood. So carbonic anhydrase is an enzyme inside red blood cells which converts carbon dioxide and water to carbonic acid and vice versa. Therefore, it's inside the red blood cell, so this is incorrect. It's intracellular. C. DNA polymerase is an enzyme that helps build DNA molecules from nucleotides. So DNA polymerase is found inside the nucleus, makes it also intracellular. Same with RNA polymerase, therefore the only correct answer is A. Number 15. Which row is correct for enzymes that catalyze reactions using lock and key hypothesis? So lock and key hypothesis theory and enzymes assumes that both the active site and the substrate is perfectly complementary at the start. They are both exactly the same size and shape. So let's see the suggestions that we have here. Effect of the enzyme on the activation energy of the reaction being catalyzed. So we know that enzymes always lower the activation energy by providing an alternative pathway with a lower activation energy. Therefore, reactions could take place at a lower energy level. Therefore, A and B is correct. Shape of the active site in comparison to the substrate. Now, changes to become complementary as the enzyme-substrate complex forms. This is incorrect. It is complementary before, during, and after the formation. This answer, though, assumes that this is the induced fit hypothesis. The induced fit hypothesis assumed that as the substance binds to the active site is triggered to change shape to become complementary, but this happens after the binding of the substrate to the active site. Therefore, this is incorrect, and the answer is going to be B. Question number 16. A scientist investigated the rate of a breakdown of hydrogen peroxide. 4 experiments were carried out using different mixtures. The results are sketched on the graph. Which row shows the correct lines for the 2 experimental measures? So, first of all, we must define our terms. A competitive inhibitor binds to the active site of the enzyme, therefore lowering the rate of the reaction. However, increasing the substrate concentration cancels out the competitive inhibitors. So as we increase the substrate concentration, the effect of the competitive inhibitor decreases and the rate of breakdown of hydrogen peroxide, as an example, reaches the optimum. Contrasting a non-competitive inhibitor which binds to an allosteric site. By an allosteric site of the enzyme, I mean a site other than the active site, completely distorting the shape of the enzyme. Therefore, no matter how much you increase the substrate concentration, the shape of the active site is completely altered. Therefore, no matter how much you increase the substrate concentration, the rate would stay constant and will not increase any further. And obviously, substrate plus enzyme only without an inhibitor would be the highest rate of breakdown. Now, what does it mean when the rate of breakdown of hydrogen peroxide increases? So, the higher the rate of breakdown, that means that the reaction is taking place the most. So, the greatest rate of reaction between the substrate and enzyme has the highest rate of breakdown of hydrogen peroxide. So, it is normal the greatest rate of breakdown of hydrogen peroxide to be the substrate plus enzyme only without the inhibitor. Therefore, 1 is going to be the substrate plus enzyme. And I've mentioned that the substrate plus enzyme plus competitive inhibitor is that if we increase the substrate concentration, it will eventually reach the optimum. Therefore, 2 is the competitive inhibitor. And 3, we know that a non-competitive inhibitor completely distorts the shape of the active site by binding to an allosteric site. Therefore, no matter how much you increase the substrate concentration, it stays constant. Therefore, this would be the non-competitive inhibitor. Therefore, these are incorrect. And lastly, for 4, it is the substrate only. So, if we only have the substrate without the enzyme, we know that the enzyme lowers the activation energy. Now, there is no enzyme to lower the activation energy of the reaction. Therefore, the reaction containing the substrate only would take place the slowest. Therefore, 4 is going to be the substrate only. Therefore, the answer is going to be B. Question number 17. Which of these substances can pass directly through cell surface membranes without using a carrier protein or a channel protein? So, for a substance to be able to pass through a cell surface membrane, it must be hydrophobic, aka not water soluble. So let's see the suggestions that we have here. We know that calcium ions must be hydrophilic because it's an ion, it's charged. Therefore, this is incorrect. For 2, it is carbon dioxide. It is an uncharged molecule, therefore it's hydrophobic and can indeed pass through the cell surface membrane without a carrier protein. Now 3, we have here a glucose. We're assuming that this is glucose. We know that glucose is hydrophilic. Therefore, it cannot pass through a cell surface membrane. Therefore, the only correct answer must be D. Question number 18. What happens to the surface area to volume ratio of a cube when the length of each side is doubled? So let's just assume that we have a cube here with a length of 1 centimeter each. So, surface area to volume ratio. The surface area would be 6, because 1 times by 1 and then times by 6. And the volume would be 1. Now, let's assume that we doubled the length and this is going to be 2 centimeters each. Surface area to volume ratio. So in this case, the surface area would be 24 and the volume would be 8. If we simplify this ratio further by dividing 24 by 8, we're going to have 3 to 1. Therefore, what happens here is that the surface area to volume ratio actually halves. Therefore, the answer is going to be B. Question number 19. Which events are part of the mitotic cell cycle? So, in the mitotic cell cycle, what happens is there is mitosis, which consists of P mat, which is prophase, metaphase, anaphase, and telophase. Then after mitosis, we have something called cytokinesis, which is the division of the cell to two daughter cells. And the rest is the interphase. In interphase, we have G1, S phase, which DNA replication takes place in the S phase, and G2. So let's see which events are part of the mitotic cell cycle. Therefore, we have interphase, we have telophase, which is part of mitosis, PMAT, telophase, and cytokinesis, which happens right after mitosis. Therefore, the answer is going to be A. 20. The estimated total number of red blood cells in the human body is 2.5 times by 10 to the power of 13. It is estimated that each day 2.5 times by 10 to the power of 11 red blood cells are removed from the circulation and are replaced by stem cells in the bone marrow. Which percentage of the total number of red blood cells is replaced each day? So, this is very simple. You just have to throw these numbers in your scientific calculator and make sure to have the brackets. So in this case, we must do 2.5 times by 10 to the power of 11 over 2.5 times by 10 to the power of 13, and then times that number by 100, and then you will obtain 1%. Question number 21. DNA polymerase catalyzes condensation reactions between molecules during semi-conservative replication of DNA. What is joined by DNA polymerase? So let's just define really quickly what is semi-conservative replication of DNA. This means that the newly synthesized DNA is going to contain one strand from the parent and one newly synthesized strand. This is what semi-conservative replication is. So what is joined by DNA polymerase? As you see in the image here, DNA polymerase joins two nucleotides, as you can see here. It joins two nucleotides together. This is a single nucleotide. A nucleotide consists of a base, whatever it is, and a pentose sugar and a phosphate group. So this is a pentose sugar and this is a phosphate group. Therefore, what is joined by DNA polymerase is an entire nucleotide. Therefore, the answer is going to be C. Question number 22. Two polynucleotide strands make up a DNA molecule. Which description is correct? A. The percentage of cytosine is 50% of that of guanine in the whole molecule. Now, as we all know that cytosine is complementary to guanine. Therefore, both of them must be in the same exact quantity because both of them are complementary. In one polynucleotide strand complementary to the other. For example, therefore, this is incorrect. B. The percentage of cytosine is the same as that of guanine in the whole molecule. Yes, this one is correct because of the rule of complementary base pairing. Therefore, this is correct. C. The percentage of cytosine is the same as that of guanine in each strand. Now, the rule of complementary base pairing occurs between two polynucleotide strands, since not a single polynucleotide strand pairs with itself. Therefore, this is totally incorrect. D. The percentage of cytosine is the same in each strand of the molecule. This is also incorrect due to the complementary base pairing because cytosine cannot bind with another cytosine. Therefore, this is incorrect and the answer is going to be B. Question number 23. The PDX1 protein is found in a wide range of animal species where it is regarded as the essential for normal metabolism. In sand rats, the gene coding for the PDX1 protein has a much lower proportion of adenine and thymine nucleotides than PDX1 genes from other animals. Finding the complete DNA sequence of the PDX1 gene in the sand rats has been difficult. Sequencing involves splitting off nucleotides one at a time from single-stranded DNA. So splitting off nucleotides, we know that they bind with each other via hydrogen bonding. Therefore, hydrogen bonds would be broken in this case. What could account for the difficulty in finding the DNA sequence of the PDX1 gene in sand rats? The PDX1 gene is present in only a small proportion of the nuclei. This piece of information is incorrect because even though that genes are smaller than chromosomes, they are still present in the entire nuclei. Plus, The only reason why it would be difficult to find the DNA sequence is due to the method of extraction. How difficult the method of extraction is, it has technically nothing to do with the location. 2. The PDX1 gene is transcribed in only some cells. So here we are dealing with genes. We are not dealing with the proteins or the products of transcription. Even though some cells only transcribe this gene due to the transcription factors, because it's the transcription factors that determines which gene is transcribed. Why, you would ask, is because all our cells have exactly the same genetic material. Every single cell has exactly the same genetic material. However, it's the transcription factor that determines the property of the cell and which genes are transcribed. Therefore, this is incorrect. All genes have the PDX1 gene. 3. The strength of the hydrogen bonding between the two strands of the PDX1 gene is unusually high. I've just mentioned previously that always the difficulty in finding the DNA sequence is within the method of extraction, and here it's informing us that sequencing involves splitting off nucleotides one at a time. Therefore, this consumes tremendous amounts of energy, and hydrogen bonds are hard to break in that strong. Therefore, this is correct, and the answer is going to be D. 4. Different tissues in a plant were supplied with a radioactively labeled substance to identify which tissues were actively synthesizing mRNA. Which radioactively labeled substances would be suitable for this experiment? Therefore, we are looking for substances which are only selective for RNA. So let's see the suggestions that we have here. 1. Adenine. So adenine is a base found in both DNA and RNA nucleotides, therefore this is incorrect. 2. Uracil. So uracil is only found in RNA nucleotides since it substitutes thymine in RNA, therefore this is correct and can be selective for only RNA. 3. Inorganic phosphate. This is incorrect because in all nucleotides we have a nitrogenous base, a pentose sugar, and a phosphate group, an inorganic phosphate. Therefore, this is incorrect. For ribose. So the ribose sugar is only selective for RNA. It's a pentose sugar selective for RNA because DNA nucleotides have deoxyribose sugar, while RNA nucleotides have ribose. Therefore, this is correct, and the correct answer must be C. 25. Which row correctly matches the structure and function of phloem sieve tube elements? So, phloem sieve tube elements are responsible for the active transport of sucrose in a plant. So, let's see the structure. A peripheral cytoplasm with no nucleus. This is correct. The reason why it has no nucleus is because the nucleus of the companion cell beside it is more than sufficient to carry out its functions. B. End walls modified to form sieve plates. This is correct, contrasting the xylem which lacks sieve plates since it's a hollow tube. C. Elongated cells joined end to end. This is also correct because it does form a tube. D. Cellulose cell wall with no lignin. This is correct. Sieve tube elements actually lack any lignin, contrasting the xylem. Function. To provide as little resistance to flow as possible. This could be one of the functions. To slow down the rates of transport of solutes. If we assume that the solutes is going to be sucrose in this case, actually it does speed up the rate of transport, so this is incorrect. C. To form a tube to transport dissolved mineral ions in water. This is incorrect. We're talking about sieve tube elements, which is responsible for the transport of sucrose. However, dissolved mineral ions in water, what transports them is the xylem. And finally, D. To prevent loss of water. So its function, as I said earlier, it's to transport sucrose, and it has nothing to do with the loss of water in xylem as an example. Therefore, this is incorrect, and the answer is going to be A. Number 26. Which feature of xylem vessel elements helps adhesion during transpiration? So the process of adhesion is when water molecules adhere or form hydrogen bonds with the cellulose cell wall. Why? Due to the OH groups in the cellulose, allowing hydrogen bond formation. Now let's see the suggestion that we have here. A, lignin forms a complete secondary cell wall. So this is incorrect. It's not lignin that forms it, it's cellulose and mainly hemicellulose. So mainly cellulose and hemicellulose. Therefore, this is incorrect. B, new vessels carry extra water as the plants grow. However, it has nothing to do with the mechanism of adhesion. Carrying extra water does not help with adhesion. Therefore, this is incorrect. No cross walls between vessel elements. This is also incorrect. D, the vessel elements form a narrow tube. Yes, we have discussed earlier that forming a narrow tube actually helps with capillary action. And it's the tendency of fluid to rise up in a narrow tube. Therefore, this does help with the mechanism of adhesion during transpiration, because transpiration is the movement of water up in the plant due to the capillary action. Therefore, D is the correct answer. 27. Mass flow is the bulk movement of materials from one place to another. How many of the vessels listed carry fluids by mass flow? So, as the definition here, the mass flow is the bulk movement of materials from one place to another, technically from source to sink. Therefore, let's see the suggestions that we have here. Let's start with artery. So, artery carries red blood cells and plasma in bulk. Therefore, this does carry by mass flow. Phloem sieve tube does carry sucrose from source to sink. Therefore, this is also correct. Vein also carries blood. And finally, xylem vessel. This is also correct because it carries water and mineral ions also from source to sink in bulk. Therefore, all 4 of them is correct and the answer is going to be D. Number 28, cellulose, lignin, and suberin are components of various plant cell walls. Descriptions of these cell wall components are listed. Which row correctly matches 3 of the descriptions with cell wall components? So let's see the suggestions that we have here. 1, a component of the Casparian strip. So Casparian strip is found in the root endoderm of a plant, and it stops water transport in the apoplastic pathway. So it blocks the apoplastic pathway completely and only allows the symplastic pathway. For those who don't know, the apoplastic pathway is the transport of water in the cell walls of the plant, contrasting the symplast, which water goes through the plant cell. Why is that? Is because there is a strong hydrophobic material outside stopping the apoplastic pathway. Therefore, the only suggestion that suits this description is going to be suberin. Suberin is the component found in the Casparian strip to stop the apoplastic pathway. Therefore, 1 is definitely suberin. 2, redirects water into the symplastic pathway. As we said, it's also going to be the suberin. So suberin is both 1 and 2. Now 3, a hydrophobic component of cell walls. So a hydrophobic component does not allow water to pass through. Could also be lignin. Therefore, 3 must be lignin because cellulose is actually hydrophilic and allows water to pass through, because as we said in the apoplastic pathway as an example. Therefore, cellulose is incorrect. 4, water interacts with this molecule in the apoplastic pathway. As we said, it's going to be cellulose in this case, and it's the transport of water in the cell wall of the plant, which consists of cellulose. So 4 is definitely cellulose. Therefore, the answer is going to be D. Shows the direction of water movement across a plant root from water in the soil to the xylem. Which arrow shows water movement only in the apoplastic pathway? So we've discussed the apoplastic pathway enough in the last question, but very briefly is the transport of water through the cell walls, cellulose cell wall of a plant cell. And the water does not enter the cell because entering the cell is in the symplastic pathway. Therefore, let's see the suggestions that we have here. So 1, 3, 5, 7, and 8 is incorrect because that is actually the symplastic pathway where water passes through the actual cell. Now let's see the other suggestions. 1 is correct. It shows water passing through the cellulose cell wall. Same with 4. So both 2 and 4 are correct. However, let's pause here for a second for 6. We've discussed that this is the Casparian strip. in the root endoderm of a plant made of suberin. Suberin stops the water transport by the apoplastic pathway. Therefore, even though there's a line here, technically it cannot pass through the Casparian strip due to the suberin. Therefore, 6 is totally incorrect. Therefore, the correct answer is going to be C. 30. Which rock correctly identifies sources and sinks of sugars? So let's see the suggestions that we have here. Root cells absorbing mineral ions. So root cells in this case is going to be the sink. The reason for that is that mineral ions pass from the soil into the roots via carrier proteins or active transport. Therefore, A and B is correct for sink. Now, storage cells of seed that is starting to grow. So storage cells of the seed is going to be the source in this case, because it is supplying a plant that is yet starting to grow. Therefore, any substances are going to be taken from these storage cells. Therefore, the answer is going to be A. The photomicrograph shows a section through a tissue with an artery, which correctly shows the type of artery and whether the blood inside the artery is oxygenated or deoxygenated. So first of all, we have to identify a few elements. It's asking for the type of artery and the blood. Now, first of all, you must identify that we're now in an alveolus. This is a photomicrograph of an alveoli. Therefore, here we are in the lung. So, know that the pulmonary artery is actually deoxygenated. Now, let's see the type of artery. So, if we zoom in a little bit further, we could see here a line in the tunica intima. First of all, you must know that the artery consists of tunica intima, tunica media, and tunica externa or adventitia. So here we can see the tunica intima made of endodermal cells. So here we could see a black line. This is called the internal elastic lamina. The internal elastic lamina consists of elastic fibers. So this is a little bit of a hint outside the syllabus. However, we know that the internal elastic lamina consists of elastic fibers. Therefore, if this were to be an elastic artery, then we wouldn't be able to distinguish that line from the tunica media, which will also have a large quantity of elastic fibers. Therefore, we would not see that line. The reason why we could actually distinguish the internal elastic lamina or this black line is because that this is actually a muscular artery. Why? Because we can distinguish between them. Why again? Is because the tunica media now has muscle fibers, and the muscle fibers are not the same as elastic fibers. Therefore, if we are able to distinguish both the elastic fibers and the muscle fibers, then this must be a muscular artery. If it's just a broad line which you are not able to distinguish between both, this must be an elastic artery because both of them are the same component. Therefore, this is a muscular artery, and the answer is going to be A. Heart surgery may cause a decrease in the transmission of impulses in the Purkinje tissue to the right side of the heart. What is a possible effect of these decrease? Now let's see the suggestions that we have here. A. Impulses would be delayed by the atrioventricular node. Impulses are already delayed by the AVN because the AVN function is to delay the impulse a fraction of a second so that both the atrium and the ventricles do not contract at the same time. So this is correct. However, it's not delayed due to the heart surgery. This is a natural mechanism in every single human being. Therefore, this is incorrect. B. The muscle of the right ventricle would contract slightly more slowly than the muscle of the left ventricle. This is correct because a delay in the transmission of impulses means the impulses reaches the Purkinje fibers or first goes down the bundle of His. So let's draw heart. First of all, from the AVN goes down the bundle of His and to the Purkinje at the base of the ventricle. Therefore, if this route or pathway is delayed, therefore the right ventricle would contract more slowly than the left ventricle. So this is correct. See, the muscle of the right atrium would not contract as fully as the muscle of the left atrium. So here we're talking about the right side of the heart. Therefore, this is incorrect. The atrial node would transmit fewer impulses. So the sinoatrial node is found in the right atrium, and its function is to initiate electrical impulses to excite the heart. Now, if the surgery were to actually damage the sinoatrial node in the right atrium, then it would transmit fewer impulses. However, this would lead for the entire heart to contract more slowly, not just the right side of the heart. Therefore, this is incorrect. And the answer is going to be B. The diagram shows a network of blood vessels that supply blood to muscle tissue in human body. What is the correct comparison between blood at X and blood at Y when muscle tissue is at rest? So blood pressure at X compared with blood pressure at Y. First of all, you must identify that this is an arteriole, this is capillary network, and this is a venule. So the earlier we are on from the artery, or the closer to the heart we are from the arterial side, the higher the blood pressure. Therefore, it's obvious that the blood pressure at X is always higher. Now, water potential of blood at X compared with water potential at Y. So, at Y, what happens? We've just discussed that this is a capillary. Therefore, fluid leaves as tissue fluid, carrying minerals and nutrients to the surrounding cells. Therefore, there's fluid lost from this capillary. Therefore, what happens is that the water potential at Y would be actually lower than X. Therefore, water potential of X would be higher. Therefore, the answer is going to be C. Number 34, scientists have shown that the oxygen dissociation curves for hemoglobin of smaller mammals are to the right of the larger mammals. What does this suggest about the hemoglobin of smaller mammals? So, when the oxygen dissociation curve actually shifts to the right, this is an indication that hemoglobin has a lower affinity to oxygen. Therefore, if it has a lower affinity to oxygen, this means that it requires a higher partial pressure of oxygen to be saturated, a particular saturation. Therefore, let's see the suggestion that we have here. A. At lower partial pressures of oxygen, it binds to oxygen more strongly than the hemoglobin of larger mammals. So here we're talking about the dissociation, not the binding of hemoglobin. Therefore, this is incorrect. B. It saturates with oxygen at a lower partial pressure. This is incorrect. If it saturates with oxygen at a lower partial pressure, then the oxygen dissociation curve would be to the left. To the left, indicating that it has a higher affinity. Therefore, it could saturate at a lower partial pressure. But in this case, which it has a lower affinity, therefore it needs a higher oxygen concentration in order to saturate. Therefore, this is incorrect. C. It releases oxygen more easily than the hemoglobin of larger mammals. Yes, hemoglobin having a lower affinity to oxygen means that it could throw its oxygen easily because the binding is not as strong. Therefore, this is correct. D. When the partial pressure of oxygen is high, it carries more oxygen than the hemoglobin of larger mammals. So the fact that it carries more oxygen as the hemoglobin of larger mammals, this means that the affinity is actually higher. However, we've discussed that the affinity is lower. So even though at the partial oxygen is high, it would carry less oxygen as of larger mammals. Therefore, the only correct answer is going to be C. Number 35. What is an effect of an increased concentration of carbon dioxide in the blood? So, when carbon dioxide increases in the blood, there is an enzyme inside the red blood cell called carbonic anhydrase. The function of this enzyme is to convert carbon dioxide and water into carbonic acid, and vice versa. And this carbonic acid dissociates into hydrogen ions and bicarbonate ions. What happens is that the hydrogen ions bind to the globin-forming carbonic acid, while bicarbonate ions leaves the red blood cell into the plasma. So let's see the suggestions that we have here. A. Increased movement of chloride ions out of the red blood cells. Okay, this is the chloride shift. What happens is that when the bicarbonate ions leave the red blood cells, what happens is that positive charges would accumulate in the red blood cell. The function of the chloride shift is that chloride from the plasma with a negative charge enters the red blood cell to balance the charges. However, this is not a direct effect of an increased concentration of carbon dioxide, since this mechanism happens all the time. Therefore, this is incorrect. B, increased concentration of hemoglobinic acid. So as I said, due to the increase of hydrogen ions in the blood, due to the increased concentration of carbon dioxide, what happens is that the hydrogen ions binds to hemoglobin, forming hemoglobinic acid. Now, hemoglobinic acid has a way lower affinity to oxygen. Therefore, what happens is that oxygen is released from hemoglobin. Therefore, this is correct. C, decreased concentration of hydrogen carbonate ions. This is incorrect. We just said both of these actually increase. D, decreased concentration of carbaminohemoglobin. This is incorrect because another way of carbon dioxide transport in the blood is it binds directly to the globin polypeptide chains, forming carbaminohemoglobin. Therefore, the correct answer is going to be B. 6. Two of the requirements of an efficient gas exchange system are a large surface area and a short diffusion distance, which correctly describes how alveoli are adapted to meet these requirements. Okay, because it has a larger surface area, this means that it actually could fold over each other, and a short diffusion distance, this means that it is only one cell thick. Now let's see the suggestions that we have here. Large surface area. A elastin fibers prevents the alveolus wall from collapsing. So this is to do with surface tension. So elastin fibers actually help with surface tension. However, this does not increase the surface area. This only prevents the alveoli from collapsing. Therefore, this is incorrect. Gases dissolve in a layer of liquid to speed up diffusion. So this is also not due to the large surface area, so this is incorrect. Alveoli are folded and interconnected. So alveoli, we just said the folding of the alveoli causes a large surface area, so this is correct. Walls of the alveoli are formed of squamous epithelial cells. So squamous epithelial cells actually does something to do with the short diffusion distance, therefore this is incorrect. Short diffusion distance. An extracellular layer inside the alveolus wall contains blood capillaries. So capillaries being very close to the layer of the alveolus is actually a reason why there's a short diffusion distance from the alveoli to the blood. Therefore, this could be correct. Alveolar walls are next to the capillaries. This is also correct. Walls of alveoli are only one cell thick. Yes, this is a reason why there's a short diffusion distance, as we discussed, due to the squamous epithelial cells. Now, red blood cells are very close to the capillary walls. This is also a reason why. However, only C contains both of them correct, therefore the answer is going to be C. 37. What defines infectious diseases? So infectious disease is a disease that could be transmitted from one organism to another, causing the similar symptoms. Now let's see the suggestions. A, symptoms are caused by a bacterium that passes from contaminated air, soil, water to a host. So an infectious disease doesn't necessarily have to be a bacterium. As we've discussed, it could be a virus, a fungus, and also a bacterium, so this is incorrect. B, symptoms are caused by a pathogen that is transmitted from one host to another. Yes, pathogen is a more accurate term we can use, therefore this is correct. C, symptoms are caused by a microorganism that is carried by a vector. So there are a lot of mechanisms of spread of this microorganism. It doesn't necessarily have to be carried by a vector. There's a lot of routes of contamination, for example, also air, soil, water, etc., etc. Therefore, this is incorrect. These symptoms are caused by a virus. This is also incorrect, and the answer is going to be B. 38. A hurricane destroys a large town on an island. People move away from the town and set up tents where sanitation is poor. Which disease is most likely to spread within a week of the change in living conditions? So here, due to the hurricane, we are looking for a waterborne disease transmitted by the contamination of water. And the only suggestion we have here is cholera. Therefore, the answer is going to be A, by penicillin. So the mechanism of penicillin, it stops peptidoglycan cell wall of bacteria forming. Therefore, it's only selective for microorganisms that actually have that peptidoglycan cell wall. Now let's see the suggestions that we have here. A, penicillin only affects host cell metabolism. This is incorrect. It does not impact the metabolism. B, penicillin only binds to the 70S ribosomes. This is also incorrect. C, penicillin only blocks mRNA. This is also incorrect. D, penicillin only blocks peptidoglycan synthesis. So this is correct, and the answer is going to be D. Number 40, which processes characterize the mode of action of phagocytes? So phagocytes are examples of neutrophils or macrophages. And what these do is that they engulf organisms participating in antigen presentation. So let's see the suggestions that we have here. 1, antibody production. This is incorrect. It's done by plasma cells. 2, receptor binding. So as we all know, how are microorganisms engulfed in the first place? Is because these phagocytes have something called pattern recognition receptors. These pattern recognition receptors are receptors that recognize foreign antigens. Therefore, this is correct. And by recognizing foreign antigens, what happens is that the microorganism now is engulfed. 3, endocytosis. Yes, endocytosis is the process where the engulfing of microorganisms takes place. Same with exocytosis. Once the microorganism is inside the phagocyte, it is thrown out by exocytosis. 5, hydrolysis. So once the microorganism is engulfed, how does it break down into multiple fragments to be presented on its surface? This happens by a process called hydrolysis by enzymes found inside these phagocytes. Therefore, this is correct, and the answer is going to be C. Thank you very much for watching this video. If you think that this channel benefits you, I'd highly appreciate it if you would subscribe to this channel and like this video. And if you have any more past paper recommendation, please leave them in the comment section down below.